to go ahead and go over the 100 Greatest Discoveries in Genetics, Episode 6 questions with you today. I've put these not due until next week, so you are going to have time to work on it. You do not have to get it in today. And after the video part where it says pick one scientist mentioned, research the scientist, write a one-page paper, I'm going to make that due at the end of next week, so you will have time to work on that too. But I just kind of wanted to go over the questions with you. You should have watched the video by now. If you haven't, you do need to watch it. It's not very long, so watch the video. And then the questions that I've posted, each one of them has like what the little section was in the video, and then it's got some questions about it. So the first one was laws of inheritance. That was Mendel, was the scientist in that. And he worked on dominant and recessive traits with pea plants. He was a monk, so he had a garden, and he grew peas out there and noticed that some of them had different shapes and colors, so he worked with that. And from that, we get what's called Mendelian traits, where recessive genes show up in 1/4 of the population. The next one, genes are located in the chromosomes. That was Wilhelm Johansen. He termed the coin genes. And then there was also another scientist with the last name Morgan that worked with fruit flies. They looked at white eyes in males and noticed that it was kind of hidden characteristics. So that's when they started to work with chromosomes and realized that females have an XX, males have an XY chromosome. And what they were noticing with that, the females could pass along this X chromosome or this X chromosome. And let's say that this is the damaged one. If you pass along the damaged one to a male, then it has to show. If you pass it along to another female, she's still got one good X chromosome, so she's not going to show the traits. We're going to talk more about this when we talk about hemophilia, but this was first noticed with fruit flies with these scientists. All right, the next one is gene control and biochemical events. That was George Beadle, and he also worked with someone with the last name of Tatum, and they worked with bread mold. And they damaged the chromosomes with x-rays and mutated them, and just looked at how they could control the genes with biochemical stuff. The next one, number 4, is transposons. That was Barbara McClintock. Barbara McClintock was not recognized for hardly any of her work. She was a very, very good geneticist, but because she was a woman, she wasn't allowed to post her information or publish it. She wasn't really allowed to talk to people about it. She did do a couple of presentations, but they weren't really acknowledged. So she did have a chip on her shoulder about that. She had a lot of challenges being a woman. She was never offered a position at the college where she was working. So she worked with corn, and she noticed that and it was maize, so like the Native American corn that has different colors. She noticed that the kernels could have different breaks in the chromosomes, and that can make them different colors. So those were called transposons. And this is how we got the idea that genes can respond to mutate to the environment. So that's with transposons. She presented those findings in 1951. It was ignored and rejected. You're going to see that a lot with the women scientists in genetics or in any field. There was a lot of women scientists. They just weren't allowed to publish the results or weren't recognized. A lot of times a man got the credit for it. Which is important right now because this is March and this is Women's History Month, so that is something to remember. So that might be something you want to write your paper on. All right, number 5, DNA carries genetic material. This was Hershey and Chase. They used viruses and a bacteriophage, which is just like a shell with DNA in it. And they noticed that it could take over. The DNA could get into the host cell and reproduce the viruses. So they actually won a Nobel Prize for their work with
bacteria and viruses and how it infiltrates the host cells. Number 6 is the double helix. That is Watson and Crick. You will always hear Watson and Crick when they start talking about the double helix. This is another one where a lot of the information was stolen from a woman scientist. The woman scientist that worked on it was Rosalind Franklin. Watson and Crick were working on the compounds, and they knew the base pairs that went with DNA. They didn't really know how it went together, though. Rosalind Franklin worked with X-ray crystallography. So she actually had some X-rays where it kind of looked like you were looking up through like a windy staircase. They had some people, they knew that she had been working with DNA. They had some people go in and get her images, and they used that to put their model together. She was not given credit for it. She didn't give them her consent to use her information that she had been working on. So they completed their model in 1962 and won a Nobel Prize. Rosalind Franklin did not, obviously. 7 is about messenger RNA. There are a lot of scientists that worked on this, so it's not one specific scientist for the answer. And what that is messenger RNA is how DNA transfers the proteins, and it'll give a little code. So messenger RNA makes a little code from a strand of DNA after it unravels. And some of the first organisms, they think, actually just had RNA in it, not DNA. So it's like the basis for life, the RNA. So they basically just discovered how it translates from DNA to RNA. RNA will make a code for whatever needs to be made in the cell. Number 8 is the genetic code. And this was Mattei and Nuremberg. They worked with amino acids and found the triplet codes. Whenever we start really looking at genetics, you're going to see that we'll write the codes out in little sections of 3. So it's a triplet or codon. And then it'll have another base that'll pair with it. This is like one side of the DNA. So that's what they were working with. And each code is for a specific amino acid that's made within the cell. So they deciphered 64 triplet codes that coded for DNA and certain amino acids. And they cracked the genetic code and made a blueprint of DNA. So their work was really, really important. 9 is about restriction enzymes. And restriction enzymes is something we use all the time now with something that's called genetic engineering. Or another term that can be used is transgenics. Or genetically modified. So you can use all of those terms interchangeably pretty much. But how you genetically modify a strain of DNA, they use something called a restriction enzyme. They found that some bacteria fights viruses by cutting its DNA so it can't replicate it, so it cuts its DNA up. It's site specific where it cuts a little strand of DNA. So they found out that there are these little restriction enzymes that helps cut DNA. And now today we use that, and it's something called recombinant DNA. It means to recombine it. So we can actually take out a piece of the DNA and insert something into it and make it a genetically engineered or a transgenic or genetically modified organism. Okay, number 10 is about RNA alternative splicing. This is in the 1980s, and this is where they were working with genes that were coding and making multiple proteins. And this is something that they use, so this alternative splicing, so working with these genes is something that they helped use to make more effective medicines, because they're looking to see where it bonds or to block certain proteins or enzymes or bonds with certain areas. So this is one way that they've actually increased the efficiency of medicines with gene splicing, so they know where everything will bond. All right, 11 is mini satellite DNA. This was Jeffries. He was a geneticist in 1984. He found a DNA variant. He called it mini satellite DNA. Basically, this is DNA fingerprinting. And you see this anytime that we're talking about DNA now, or if you watch any crime shows, it'll show like the person's DNA and what it looks like, and then they try to find the match. So you'll have your little DNA thing, and you know, you've seen that where it's got the little lines on it. This is what they call mini satellite DNA. So it's just like a little snapshot into the DNA. And they can look at it, and if it lines up, then you can tell that that was the person whose DNA it was. Or you can look at it and say, yeah, half of these are identical to this person, so this person has to be a child of that person. And so we use that now to help solve a lot of murder cases and crimes that have went unsolved. If we can find DNA from the suspect, or if they have DNA still in evidence, then they can go back now and use this little mini satellite DNA. The machine's called a gel electrophoresis, and it'll just make these little lines in it, the pattern to look at the DNA. All right, 12 is about RNA interference. That was in 1997. That was Fire and Mello. They made synthetic RNA and put it into a roundworm. And there was a mechanism in the worm that actually destroyed the RNA. So they were just seeing how the body would respond to synthetic RNA. And then 13, the one that says 25,000 genes. This was a large project, one of the largest efforts in history by a scientist to map the human genome. So at first it was a bunch of different scientists working on it, and they were all like in a race to try to be the first one to do it, but actually they ended up coming together and making it one like cohesive unit to work on it and to get it done. So in June 2000, they broke the genetic code, and they sequenced the human genome. So the entire DNA sequence, they completed it. And from there they went on and sequenced all kinds of other animals. And so from there, now we're using that information for a multitude of things in science and in medicine, like gene therapy, diagnosing people. We can tell where genes are mutated, or where there's a recessive trait in there, like for cystic fibrosis or for hemophilia. So there are markers that we can look at now because we have this Human Genome Project where it mapped out the entire human genome. Okay, so you can answer these questions if you want to go ahead and do it. If I do want you to watch the video if you haven't before next week. I will give you some time if you are in class to work on these. And if you want to go ahead and get started on the after the video, the scientist, pick one of the scientists mentioned, research the scientist, background info, short biographical info, their work in genetics, like where they studied, where they did the research, what they worked with, what they found, how old they were, if they received a Nobel Prize or any recognition, and when their studies took place. Write a one-page paper on your scientist, no copying and pasting. List your sources at the end of the paper that's not included in the one page length. Put it in a Google Doc, save as a PDF, and attach it in this assignment. So, and like I said, this is Women's History Month, so you might want to think about doing one of the women that was mentioned. If not, that's fine. There's a multitude of scientists in there that you can choose from. But there's the rundown answers to the questions, so you can use that and go ahead and get started on that, and then we'll finish it up next week.